Okay, let's go ahead and get started. Welcome everyone to Kate Montgomery's Thesis Defense. My name is Scott Delp. I've been Kate's advisor. It's certainly been a pleasure. So Kate came to Stanford about 6 years ago from Rice. She was a biomedical engineering major there, and I still remember interviewing Kate. She came into my office, and like all the other interviewees, super bright, and Kate's very creative. But what struck me and some of the other interviewers is that she's also very determined, which if you know Kate, you know that's one of her key characteristics. One of the nice things about being a professor at Stanford is that we get to work with truly outstanding young scholars. And to see Kate come in with somebody with great potential, with really great intellect and creativity and drive, and turn into a truly outstanding scholar, neurotechnology innovator, bioengineer extraordinaire has really just been a pleasure for me. One of the things that you'll notice from her work, it's characterized by teamwork. There are many people who have collaborated with Kate, and she has really been instrumental in bringing teams together. Folks from my lab, the Optogenetics and Neuro group in my lab have been great supporters, and also to bring together people from Carl Deiseroff's lab, Ada Poon's lab, and other people around campus has been a real pleasure to get to know everybody through Kate's collaborations. So without further ado, Kate, show us what you got.
Great. Thanks, Scott. Let me just turn this guy on. Okay. So thank you, Scott, for that great introduction. It's so wonderful to see so many friendly faces and so many new faces out there. I'm excited to tell you about my work that I've been doing over the past 6 years. So I guess without further ado, I'll get started. Okay. So my work has been important in bringing together tools for optogenetically controlling neurons outside of the brain. Now, the spinal cord and the peripheral nervous system are extremely important for mediating our interaction with the world. And optogenetics is this revolutionary neuroscience tool developed a few years ago that has great potential to give us insight into how these neural circuits work. So this optogenetic toolbox that I've helped develop as part of my thesis work, it's been focused on a couple of major neural systems, control of pain and motor control, and in addition, creating these very small wireless light delivery systems. So to give you an outline of how this presentation will go, I'll start by discussing some of the background of optogenetics beyond the brain, and follow up with control of pain, where we were able to demonstrate being able to control these neural circuits of pain using optogenetics, both inhibition and stimulation, as well as how we were able to control motor circuits in an awake and freely moving rat, and finally end with wireless light delivery systems for controlling circuits throughout the body. And at the end, I'll wrap up and open up the floor for questions. One of the reasons we were motivated to study these peripheral circuits, these circuits outside of the brain, is that there may be some very important clinical translation applications for these systems. For example, using optogenetics in the periphery would probably be less risky because it heals more easily and it's easier to access. And there may be some lesser regulatory burdens for hurdles for overcoming this. But before I really get started, I'm going to just introduce you to optogenetics for anyone who is not familiar with the technique. Optogenetics is a way to control neurons with light-sensitive proteins called opsins. And an opsin, this light-sensitive protein, one example of which is channel rhodopsin 2, which I will frequently call CHR2, is found in an algae called Chlamydomonas reinhardti, which is found in soil and fresh water. This organism has this protein in its cell membrane called channel rhodopsin 2, which is an ion channel activated by blue light. If we were to express those opsins in a neuron instead of algae, it causes the neuron to be depolarized, which typically stimulates the neuron. Halorhodopsin, in contrast, is an inhibitory opsin, or NPHR, found in these haloarchaea, which are typically found in these very salty regions. This haloarchaea is an extremophile, and it uses NPHR as a chloride pump to pump chloride across the cell membrane. When expressed in a neuron, this is hyperpolarizing or inhibitory. So with these two proteins that I've used in my PhD work, we're able to both turn neurons on and turn them off. But how do we actually get these proteins into neurons? There's many ways that other researchers have used, but our group in particular has focused on using viruses. So to do this, we take the DNA of this opsin plus this protein called yellow fluorescent protein, which allows us to see the opsin underneath the microscope. You'll often see in the experiments that I'm about to show that we often have a YFP control, which is just the DNA of the YFP alone. We package the DNA into a virus called adeno-associated virus, or AAV, and then we inject it into the region of the nervous system that we want to control. Now, this technique of optogenetics has been revolutionary to neuroscience since its introduction in 2005. And today, hundreds of articles are published every year using optogenetics as a technique. And it's used to study almost every major brain function or pathology that you can think of. Now, using optogenetics to study regions outside of the brain in mammals of intact tissue has been less explosive here. But if you zoom in on this y-axis, you can see that it's actually really starting to take off. And using optogenetics to investigate these circuits really could offer some great potential for learning more about the systems that are mediated by the spinal cord and peripheral nervous system. So some of the challenges that might be hindering some of this translation of this peripheral optogenetics, there's a few that I want to talk about. The first is this target variability. Not just neurons are electrically excitable. There's other types of electrically excitable cells, such as pancreatic cells and muscle. So being able to create an entirely new system to hit each of these different targets may be a challenge and can hinder development. Additionally, there may be broader areas of illumination that are required. Tissue may be more opaque in the periphery, and there may be electrical noise and motion artifact noise contributed by being in the periphery. Some of the neurons in the periphery are the longest neurons in the body, and obtaining sufficient expression throughout the neurons can be challenging. And importantly, outside of the spinal cord and brain, there's a ramped up immune system. It's not as immunoprivileged as the brain and spinal cord have the advantage of being. So when you express these foreign proteins that we induce with a virus, it may be attacked by the immune system and regulated to be not expressed. And finally, this is important too, the movement of the animal is more extreme outside of the brain. In the brain, you have this advantage of having a relatively stable brain against the skull, which the skull offers a great location to anchor some sort of medical device. But you don't have that advantage in the periphery. Despite these challenges, we and others have imagined various targets that could be optogenetically controlled. These include sensory and motor circuits in these nerves and spinal cord, as well as controlling sensation through peripheral nerve terminals in the skin. Additionally, people have used optogenetics to kill metastasized cells in the periphery, as well as to imagine better tissue engineering strategies for hearts by controlling cardiac muscle directly. Some people have even used optogenetics to directly control skeletal muscle and to induce glucose tolerance in diabetic mice by controlling pancreatic cells directly with optogenetics. In my projects, we were able to control peripheral pain neuron terminals as well as motor neuron axons within a nerve. And finally, we're able to target diverse regions throughout the animal's body using wireless light delivery. This first project I'm going to talk to you about is this control of pain. The reason that we wanted to study neuropathic pain is one example of one of the pain conditions that we wanted to investigate. It's because this type of pain is extremely debilitating. Neuropathic pain is characterized by damage to the nervous system, which over time, can develop into a pain as a disease. Pain initially can be a symptom of a normal process, but when it persists, it becomes extremely debilitating. Some of the symptoms include light touch, evoking burning pain, as depicted with this hand right here, but other people have also reported tingling and numbness. This type of pain in particular is very resistant to traditional pain therapies and affects a pretty large percentage of the population, about 1%. So we wanted to investigate how we could use optogenetics to control these pain circuitries that may be affected in neuropathic pain. And this project was conducted, co-led with Srivat Zayur, who helped me with this project as well, and we published this project last year. Before I begin, I'm just going to introduce you to nociceptors, or fibers that transmit pain information from the periphery. And we were able to target these nociceptors with a simple nerve injection. We injected this virus that I told you about, CHR2-YFP, into the nerve. And we were able to see, after injection, that we had indeed targeted the fibers that we had meant to target. Right here is a cross-section of a nerve. So all those little circles you see are axon cross-sections. And you can see some of these are myelinated. This purple right here is a stain for myelin. And we expected that our unmyelinated nociceptors would not have myelin sheaths around them. And you can see that here, that there are no circles of myelin around them. So we had effectively targeted these unmyelinated fibers. But what did that mean functionally? That's just a histology picture. Did we actually functionally express CHR2 in these pain neurons? So we took animals that we had injected, and we shone blue light on their paw. And this was a really exciting experiment to do, to see for the first time that we were indeed stimulating these pain neurons through the skin. Of course, there's an important control to do here, which is to make sure that it's not a function of non-optogenetic stimulation, that it's not just the light that's causing this effect. So we shone yellow light on that same animal, and the animal didn't seem to mind. The next one is using this YFP control. This mouse, we shone blue light on its paw, and the blue light alone was not the effect. So we quantified this effect, how quickly the mouse withdraws its paw. And you can see here at around 20 seconds, eventually the mouse will just walk away and move on for non-pain reasons. But when you increase the light power density, you see around this 1 milliwatt per millimeter squared, the mice pretty regularly lift off their paw. We weren't sure what was going on in this region, however. The withdrawals were less consistent. So we wondered what was happening here. To do this, to understand what kind of pain processing was happening at these lower light levels, we constructed a floor that a place preference chamber where one room was lit with blue lights on one side and red light on the other, and the mouse could freely move between the two rooms and choose which room to stay in. We could measure how long it stayed in each room. When we had these YFP control mice, it didn't matter if the light was off or on. They spent about 50% of their time in the blue lit room. And this was true of CHR2 positive mice with the light off. But when the light was turned on, they dramatically decreased their amount of time that they spent in this blue lit room, indicating that even at these low light power levels, we were affecting pain processing. We also wondered how this low level of light power would affect mechanical thresholds or sensitivity to touch. We found that we could shine these special LED arrays. This test right here is called the von Frey test, and it's not typically set up to work with optogenetics, so we had to figure out a way that we could use optogenetics with standard pain assessment tools. So we created this LED array and shone this low level of blue light while we applied some sort of mechanical stimulus. And we found that wild type or control mice had the same mechanical threshold whether they were exposed to this low level blue light or not. Whereas CHR2 expressing mice were more sensitive to touch when the blue light was shown on their paw at the same time. Indeed, the same thing happened with thermal thresholds, in which the the control animals and the CHR2 animals without blue light were all around this normal thermal latency, but then with blue light, they became more sensitive to heat and withdrew their paw faster. Overall, we were able to demonstrate that we had excitatory control of pain and could improve future experiments where we could find out how C fiber activity contributes to chronic pain. But knowing how we can stimulate these fibers is really only half the equation, especially if we want to know more about developing therapies. In a therapeutic development, we want to inhibit pain, not stimulate it. So to do this, we used this NPHR halorhodopsin that I introduced you to at the beginning, and we used it to optogenetically inhibit pain. We used the same similar kind of injection into the nerve where we just changed out the DNA that was injected. And we were able to show that we could increase thresholds using yellow light. And here you can see the mechanical threshold increases and their thermal threshold increases. These are regular mice. They don't have any kind of pathological pain. What would happen if we tried to inhibit pain of an animal that's experiencing pathological pain? So to do this, we used a chronic constriction injury, which is a very well-used model of neuropathic pain to understand how it works in humans. And to do this, we tied a single suture lightly around the nerve, and over time, edema and compression would damage the nerve fibers. Within a couple of days, mice exhibited pain-like behavior. And you can see here this pain-like behavior. We measured mechanical thresholds and thermal thresholds of mice, both before injury and after injury. And you can see that they become more sensitive to touch and more sensitive to heat. But when we shine yellow light on them at the same time, we can reverse those thresholds and return them back up to pre-injury levels. One of the things that we came across while doing these experiments was that we found that expression of CHR2 and NPHR didn't last for very long in some animals. And in fact, most of the time it decreased at least a little bit. And what we were thinking is happening is this immune shutdown of opsin expression, one of the challenges of working with optogenetics in the periphery. You can see here in this picture DAPI stain, which shows cellular infiltration, which is much more increased than over here. These are probably immune cells, and this IBA1 stain is a stain for microglia, a type of immune cell that's also infiltrated here. If we were to take this picture and look at it 8 weeks ago before here, you'd see green cells lighting up here, but now you just see a little bit of green left over. That's the CHR2 YFP that we are trying to express in these cells. So this expression has decreased over time. If we want to be able to do longer experiments and someday be able to clinically translate optogenetics, we'll need to find a way to express these opsins for longer periods of time. Some of the potential solutions to explore this issue is to both improve the tolerance of the body to these different viruses, perhaps using a different viral vector to induce expression, maybe a different vector that's less immunogenic, or modifying the immune system itself by using immunosuppression, for example. Overall, optogenetic control of nociceptors may improve therapies for these clinical pain problems. One of the easiest ways to imagine that is directly applying this as a therapy in itself. The way that we showed how we could control these pain fibers was through a simple nerve injection and shining light through the skin without the need for an implant, which would significantly reduce the regulatory hurdles. In addition, we were able to show reversal of thresholds in a model of neuropathic pain and bring the animal's mechanical and thermal thresholds back up to its pre-injury levels. But in the meantime, this technology enables research of pain and sensation, and we can learn how C fibers contribute to spinal circuit organization and how C fibers in activity in different pain models contributes to their development and persistence. So in this last project, I described to you how we were able to control pain through the skin of peripheral nerve terminals. But there's many kinds of neurons that don't actually project to the skin. For example, in the nerve, there's motor neurons that never make it to the skin. How would we deliver light to a region like that? For this next project, I'll show you how we were able to control motor neurons deep within a rat's body and be able to specifically control those neurons. One of the reasons we were interested in controlling motor circuits is that disability in motor control can be quite debilitating and is quite common. Any kind of paralysis affects approximately 2% of the population, and it's caused by many different diseases and injuries, such as stroke, spinal cord injury, and multiple sclerosis. In extreme cases, electrical stimulation can be used to reanimate limbs and muscles that have been affected by paralysis. But this type of stimulation is quite fatiguing, and coordinating multiple small muscles to work together can be quite challenging. In 2010, Mike Llewellyn, a grad student in our lab, was able to show that optogenetic control of motor neurons is less fatiguing than electrical stimulation. As you can see here, muscle tension or force of muscles decreased rapidly with electrical stimulation, whereas with optical stimulation, it stayed quite high. However, this research was done in an anesthetized transgenic animal, meaning that it's a little harder to imagine how it could be translated to humans someday. In fact, this cuff right here is quite rigid and not implantable long term. How we can change that system into a system that can be put into an awake and freely moving animal was one of the motivations for doing this research. This project was completed co-led with Chris Town, who I see in the audience over here. This project was really exciting to work on, and we were able to do some pretty cool stuff here. So in this project, we used AAV6 CHR2. It's the same virus that I told you about in the pain neuron work just a few minutes ago, except instead of injecting it into the nerve and transfecting C fibers, we injected it into the muscle and infected motor neurons and not sensory neurons. So this is an example of viral tropism. This virus has this curious property of retrograde transport when injected into the periphery, where it can be taken up by nerve terminals and moved backwards and expressed forwards from there. And we were able to show here that when we inject two different muscles, this GN, gastrocnemius muscle, and TA, tibialis anterior muscle, we could independently get these two different motor neuron pools. So this is just a dye here, where this is the green AAV6 here. And you can see that these motor neuron pools do not overlap. What we imagined this meant is functionally, if we shine light on the entire nerve bundle that contains both these red neurons and these green neurons, that we would only activate the green neurons, only activate the muscle that we had injected. And that is exactly what we saw. You can see here how when you stimulate with an electrical stimulation cuff, you stimulate all of the motor axons within that bundle. Whereas if you stimulate with optogenetics, you only get your targeted muscle, and the other muscles stay silent. So we were able to show we could do this targeted control of motor neurons. But how do we do this in an awake and freely moving animal? We need some way to deliver light to the nerve. And this wasn't this was not an easy task to accomplish. It took about a year to even figure out how we could do this, because this had to be both flexible enough to move with the animal, but strong enough to not break over time and not damage the nerve in the process. So this strategy that we developed is by using this very flexible fiber optic that is connected to this ferrule at the top, which is cemented to the skull. This ferrule can be connected to some patch cable that is then tethered outside of the animal whenever we want to control those neurons. The light is delivered through this fiber optic down to this polymer nerve cuff that wraps around the nerve. When the animal has this on, it can freely move around. Up in detail, this cuff right here, you can see that we embedded this aluminum foil within the cuff so that any stray light could reflect back to the nerve if we didn't hit it straight on. You could see the fiber goes all the way through the cuff and all the way through the foil to hit the nerve within. And this whole device would be tunneled underneath the skin of the rat. So we wanted to know, does this actually control these motor neurons and while the animal's walking around? So we trained our rats to walk on these treadmills right here. And that was not as straightforward as you would think either. But we trained the rats to walk on here, and we implanted these electrodes, these EMG electrodes, to measure their muscle activity. We implanted one on each side of the body. And then we implanted this nerve cuff as well. And you can see here, this is electrical activity of the muscles. And you can see this left leg and right leg, and you can see it walking. This is physiological, left, right, left, right, left, right. But when we shine light, shown by these black tick marks right here, you can see how we were able to stimulate the muscle optogenetically. So we can superimpose our own electrical activity on top of the physiological activity, which really offers us a lot of potential someday for modulating gait and potentially using closed loop control someday. And this is a zoomed out trace of this too, where you can see the stereotypical muscle activity. Overall, we were able to show optogenetic control of motor neurons in the periphery, and being able to do it in awake and freely moving animals removes the confounds of anesthesia and allows us to study muscle and motor function without interfering with anesthetics. We were able to target to our muscle of interest, which means someday we may be able to use multiple different colors to do reanimation of limbs that is very controlled. And overall, future directions of this project, as we showed in the pain inhibition, motor neuron inhibition will be extremely important. This is motivated by treatment of spasticity and other hypertonic motor disorders in which there's no real good treatment for inhibiting this excessive motor activity. And it has some really important advantages over electrical stimulation. There's additional potential for clinical translation and new research in terms of learning about spinal connections with sensory circuits, as well as learning about the protective or rehabilitative effects of motor neuron stimulation and how it can be used to heal the peripheral nervous system when it's been damaged by injury or disease. So far in this project, I've described to you our work to target pain through the skin and motor circuits through the nerve. But these light delivery systems, although they work quite well for our particular targets, we wanted to know how could we develop a light delivery system that is broad and adaptable and can be used to target almost any circuit that we want to hit. And we imagined that this strategy would only work with some sort of very small wireless device. This project to wirelessly control circuits throughout the nervous system was imagined by Ada Poon's lab and her group. And I worked directly with Alex Ye, who also co-led this project with me, to wirelessly control optogenetically circuits throughout the nervous system in mice. We were motivated to do this research because current wireless optogenetic systems are quite large. As you can see here, this is pretty typical of a wireless optogenetic system, which is, if you look at a scale of mass right here, these are some of the other wireless optogenetic systems, and this is the size of a mouse head. So they're about the approximate mass of a mouse head. Whereas the devices that we were able to come up with using Ada Poon's technology are much smaller. They're approximately 2 orders of magnitude smaller than the previously developed optogenetic systems. And this is, if you want to imagine the size of it, about the size of a black peppercorn. Now, there have been multiple studies where they've incrementally decreased the size of these wireless optogenetic systems, but when you decrease it by this much, it really brings into play a few key features. First of all, having devices this small means that you can implant them outside of the brain. You can attach them to a tiny nerve within a mouse or attach it to the spinal cord. For people studying the brain, being able to implant these devices on the skull means that you can do things like social behavior, where the animal is not too impacted by having such a large device on its head, as well as being able to move through obstacles and enclosures freely. Right here you can see a mouse running on a wheel with an implant on its head. And maybe most importantly to behavioral neuroscientists is that the device is fully internal. And this is important because it means the animal doesn't need to be handled before experiments, which can be quite distressing to animals and confound especially studies of things like anxiety. It also enables chronic experiments where you can optogenetically manipulate these circuits without human intervention at all by having the wireless power going constantly. At the root of all wireless transfer is how we create this electromagnetic field to power a wireless device. And here we use something called a resonant cavity developed by the Poon Lab. And this resonant cavity is, the physics of it are described here in this paper. But briefly, we send a signal into the resonant cavity, and energy, for the most part, is contained in this hollow cavity, except for exactly where the mouse is on top of the cavity. In this way, energy is transferred to the mouse, but only where the mouse is, which makes this energy transfer extremely efficient and self-tracking. Importantly, these wireless devices can be built without any special facilities. Some of the, for example, some of the tools required are a hot plate or a dissecting microscope. To validate this device, we tested it in 3 different regions of the nervous system. The first I'm going to describe to you is implanted into the brain. To do this, we wanted to control the motor cortex, so we implanted this device such that the coil and the board were above the skull, but thin, twisted wires were pushed into the brain to the circuit that we wanted to control, and a tiny micro LED at the very tip illuminated the region of the brain we wanted to control. To stimulate this motor cortex, we wanted to measure the animal's behavior upon stimulation. So we measured the activity of mice in this circular chamber before light was turned on and after. And you can see that a mouse that is just wandering around without its light turned on has no particular direction that it's going. When we turn on blue light, however, the animal circles in a dedicated way. This is perhaps most easily illustrated by showing the change of speed that the mice exhibit when the motor cortex is stimulated. You can see that with the light off, they move less than when the light is on. And this can be reversed. So after we turned it off, they went back to their baseline levels, and they increased their speed again when the light was turned on. Secondly, we wanted to control some area of the nervous system that has been quite challenging up until this point. So we wanted to control the spinal cord, which has important implications for understanding motor circuitry and sensory circuitry. And to do this, well, so far, there's only one method currently published about how to control these spinal cord circuits, and it involves fusion of multiple vertebrae and involves substantial disability in the way the mouse can normally walk. And this is with a tethered fiber optic. So we wanted to create this wireless system to control these spinal circuits. To do this, we drilled a tiny hole in the vertebrae and cemented this implant directly to it. You can see in this version of the implant, there's no extension that goes down into the nervous system. It's just an LED attached to the board here. And here you can see this mouse that has the device on, that the implant is totally underneath its skin. To verify that we could effectively stimulate spinal cord circuits, we shine light on the spinal cord, and here you can see a cross-section of the spinal cord, of the top surface or dorsal surface of the spinal cord. And these nerve terminals that are coming in right here are expressing CHR2. When we shine light on these nerve terminals, we see this increase of CFOS expression, these little magenta dots here. Now, CFOS is a marker of neural activity. So after stimulation of these circuits, we would expect to see an increase of CFOS expression in neurons that have been recently turned on. That same level of increased expression did not happen in the non-stimulated side of the spinal cord, nor did it happen in a YFP control animal that was not expressing channel rhodopsin. These results can be summarized across multiple mice here by seeing how asymmetry of this CFOS expression occurred in the CHR2 stimulated animals. And finally, so far I've done brain and spinal cord. We also wanted to be able to control peripheral neurons. So to do this, we used the same pain mouse model that I described to you at the beginning, where their nociceptors are expressing channel rhodopsin. And we took this implant that's been modified for the peripheral nervous system with this very long extension so that we can implant the board and coil up high and tunnel this lead or extension down to the area that we want to stimulate peripheral nociceptors. And you can see here that since the light is shining out through the skin, it's quite bright. To understand how this affects mouse behavior, we constructed a place preference chamber that's similar to the one that I described at the beginning, where instead of the floors being lit up, however, under one of the floors we placed this resonant cavity, and under the other we placed this support structure. We allowed the animal to freely choose which room it would stay in. And these are just YFP mice. You can see that they spent about the same percent of time in this resonant cavity chamber, whether the light was off or on, except you may be wondering what happened to this mouse right here. Why is it spending so much time in the resonant cavity chamber? And the truth is that it fell asleep, and such is the perils of behavioral neuroscience. But CHR2 animals, the important ones here, well, both are important, but the ones we wanted to see what their behavior would be, they decreased the amount of time they spent in the resonant cavity chamber when the chamber was on. So we were able to demonstrate that we could induce an aversion to having their pain neurons turned on with those wireless implants, replicating the place preference behavior that we saw in the first transdermally illuminated study. So some of the next steps that we could imagine for moving this research forward about wireless optogenetics is creating resonant cavities for other behavioral setups. So far, we have a resonant cavity that's about 21 centimeters in diameter, which is great for many types of studies, but we would also want to know, could we create these resonant cavities for larger animals, such as rats, or create large area tasks, or resonant cavities for large area tasks like this Y maze and other mazes. You could also imagine something that would allow us to wirelessly control these implants in a home cage as well. One of the great advantages of this miniature wireless light delivery system is that we can imagine being able to adapt this technology to target other types of circuits throughout the body, so nerves or maybe even organs or other terminals throughout the body. Importantly, we are hoping that this device has minimal influence on animal behavior and that it is also fully internal, which means that we don't need to handle these animals and will in the future enable chronic control of circuits. So overall, we were able to show this bidirectional optogenetic control of pain, meaning we could both turn on and turn off pain. And this was done with just a very straightforward nerve injection and being able to shine light directly through the skin. This optogenetic stimulation of motor neurons we were able to achieve it in this freely moving animal that's walking without damaging its nerve, and by being able to control specific motor neurons without affecting the other ones. And finally, we were able to develop this miniature wireless light delivery system that's 2 orders of magnitude smaller than other wireless optogenetic systems and will allow for control of multiple neural targets and potentially even non-neural excitable cells throughout the body. At this time, I'd like to thank the organizations and the people that have made my research possible, including BioX, who not only funded the snacks that are available over there, but also helped build the building that I work in and that we're sitting in now, funded many of the projects that I've worked on, and also funded my stipend and tuition for the last 6 years. So I'd like to thank them, and as well as the NIH, who funded some of these projects as well. I'd like to thank my advisor, Scott Delp, for being just an incredible advisor. He's always pushed me to do things that are just on the edge of impossible. And I also would like to thank Scott for encouraging me to wear a helmet, because that came in really useful last year when I went over my handlebars. So Scott is both an incredible advisor and mentor as well. He shows that he really cares about us. I'd like to thank Carl Dijsselroth and Ada Poon, who have been my colleagues and collaborators, but have also been extraordinary mentors to me. And it's been such a pleasure working with both of you and learning all of the things that you have been so wonderful to teach to me. I'd like to thank Xiang and Rog Malenka for being on my committee and for being here today. I'm really excited they have incredible neuroscience and clinical experience, and I'm excited to have some great conversation with them in the closed session. Okay, I deserve a huge thanks to my teammates who co-led these projects with me. Srivat Iyer, he's one of the most incredible scientists I've ever worked with, and I'm so honored that I have been able to work with Srivat for the last 6 years. Srivat has contributed to every major project in this dissertation, and I'm so thankful for him. I'd like to thank Chris Town, who really invigorated our lab and breathed life into our lab with his extraordinary expertise in both gene therapy and animal work, but also with his cheeky Australian charm. And he really brought us together as a team, and it's been such a pleasure working with him. I'd like to thank Alex Ye, who was the talented electrical engineer who helped turn the theory of wireless power transfer into tiny buildable devices that we could put into animals. Working with Alex has been such a privilege, and even though we spoke a somewhat different language at the beginning of our project, we really grew to be great friends. I'd like to thank all of my collaborators, though. All of these projects require an extraordinary amount of teamwork. People in the Dijsselroth lab, Charu, Emily, Logan, Sue, have been so incredible to work with, and it's been fun to run into you guys in the basement. I'd also like to thank John, Yuji, and Vivian from Ada Poon's lab for their contributions as well. And I'd like to thank the entire Nimble Neuro team, that's the subgroup of Scott Delp's lab, that we work together on these optogenetic technologies and other neuroscience applications. And both past and present members have been really influential in my development at Stanford. Amy, Chris, Holly, Helena, Sam, Sarab, Andrew, and Stephanie have been really wonderful. I'd like to thank the entire Nimble lab for being both incredible colleagues, and they've been not just colleagues to me, though, they've been incredible friends, and they make it possible to go out for a midweek outing or up to the slopes and enjoy each other's times as friends. I'd like to thank all of my friends, especially Jenny, Shannon, and Matt, for their support and endless energy and enthusiasm. And finally, I'd really like to thank my family. My family has been so supportive for me. Many of them are out in the audience today, and that's so incredible. I'd especially like to thank my parents, who have been so supportive and call me every week and really make doing the thesis and the dissertation so much more fun. Okay, at this point, I would like to open the floor for your questions. Jenny?
Okay, so I'm going to ask the question that everybody wants to know. How long do you think it'll take to get optogenetics to have actual clinical uses in humans?
Yeah, that's a question that many people have different answers to. So the first question is, how soon will it go into preclinical trials? or go into clinical trials, I'm sorry. So I think that could be quite soon, maybe within 4 years. So one of the first applications that I haven't described here is perhaps using optogenetics to control the retina and help work with applications in blindness, things like that. Charu?
Have you measured the light power coming from your LED devices? Is it enough?
Right, we can get up to 20 milliwatts per millimeter squared. So it's sufficient to do optogenetics with that. Mm-hmm. Sam?
The more you ask, the less we add.
So you've shown that you can go into brain, spinal cord, peripheral nervous system.
Sure.
Can you multiplex in one animal? So can you control? Are you able to control like specific devices, or do you need different?
Right. Yeah, so the current design of the device is quite a simple circuit, and it would be without some sort of circuit control to individually control different devices. We'd need a more sophisticated circuit on board. But right now, you could potentially turn on a couple at the same time, but the power that each will take will slightly decrease. So it would be something that would have to be characterized if you want to use multiple implants in the same animal, or even multiple animals within the same cavity. Yes?
Curious, for the peripheral nerve experiments, tell us a little bit about the exploration of the different AAVs that you went through and how you got to target the C fibers without a specific promoter.
Sure. So we had the benefit of having Chris Town come into our lab, and he had already done some preliminary research showing, just in a gene therapy sense, not with opsins yet, that AAV6 could infect these nociceptors when injected peripherally. But in addition, we did inject multiple types of AAV serotypes to screen for which ones we could get certain kinds of specificity. We tried AAV8, AAV2, and we got different patterns of infection in different types of neurons. And it seemed AAV6 seemed to be quite specific for these nociceptor-like neurons when we looked at histology of them. So it seemed a promising avenue from that point of view. Jen?
Thinking about applications either to research or clinical use eventually, something that seems important would be like how much control you have on the amount of pain you're either producing or inhibiting. Do you have a sense of how closely you can control pain?
Right. So the answer to that is we can increase the light power and increase you can see like the light power increase up to a certain point, you get faster and faster flinch responses. Knowing exactly how much pain you're inflicting, though, has been a problem with pain neuroscience from the beginning, because we don't know with a viral injection what percentage of those neurons are expressing CHR2 before we do histology. But yeah, I guess after histology, though, you can have a better idea of how many neurons have been infected. So we haven't been able to quantify exactly how much pain they feel.
How chronic have you implanted these wireless electrodes? Months, years?
Sure. Not years. So for ethical reasons, we do our experiments quite quickly. For the experiments shown here, the next steps would definitely be figuring out exactly how long these devices last in animals. And these devices would need to be optimized for that, special biocompatible coatings that are made to last for long periods of time. We have been able to have functional implants for up to a month or two after implantation, but that wasn't a carefully controlled experiment. Did I see a hand? Yeah.
Can you have both inhibitory and excitatory opsins on the same motor nerve?
It is possible. There is a construct that links the two together, so you can have both halorhodopsin and channelrhodopsin in the same construct. Unfortunately, I think that it's a little harder to express those opsins together. And in particular, you may have noticed in the optogenetic control of motor neurons, we weren't able to get inhibition of motor neurons. It's been quite a challenging thing to get halorhodopsin to express efficiently and effectively in motor neurons. So I'd anticipate in motor neurons it would be quite a challenge, but maybe a different inhibitory opsin would work better.
Yes, it would be useful to get rid of the antidromic pulse and
Absolutely.
ensure inhibition.
Yeah?
Have you looked into non-viral means of transfecting the cells?
Sure. So there's non-viral means. I mean, there's transgenics, which animals that are born with opsin. Electroporation is an option, but it can be damaging to the cells. There's one way of using naked DNA transfer, which doesn't use viruses, but that usually doesn't last very long. So there's other options that we've considered, but we haven't actively tried with experiments there.
Good. Let's thank Kate for a great talk.